So here's an update for the channel. We're going to be changing directions of how the content is made. So I used to do content based on just facts and knowledge and then make that into a video. But for now on, I think I'm going to actually read a scientific article and then inform you guys about the key points and the results. And if you like this kind of video, please comment below so we can make this a permanent thing. But the main idea here is we want to show how interesting science really is. And welcome to the Survival Guide. Today's topic we're going to be reviewing is actually an article review. That is Alcohol Abuse in Pregnant Women, Part 1. We're going to be looking at the effects on the fetus and the newborn, as well as the genetic susceptibility to alcoholism and behavioral developmental effects due to prenatal alcohol exposure. Here is the source that we'll be using to uncover this topic, and just here as a reference. The introduction to prenatal exposure to alcohol. The ingestion of alcohol during pregnancy can lead to developmental delays and behavioral changes. Ethanol can affect the fetus in a dose-dependent manner. Ethanol is able to affect the central nervous system, CNS, throughout the entire pregnancy. High repetitive doses of ethanol during pregnancy can lead to a 6 to 10% chance of fetal alcohol syndrome in the offspring. Low repetitive doses of ethanol can lead to alcoholic-like effects, and this can lead to growth disturbances, behavioral changes, and a slight intellectual impairment. A genetic susceptibility to alcoholism. Ethanol can be metabolized using the enzyme alcohol dehydrogenase, also known as ADH in this article. This can lead to the production of a toxic intermediate product known as acetaldehyde. Increasing ethanol consumption can lead to increased levels of acetaldehyde. Acetaldehyde may increase if a person has a polymorphism for the loci encoding alcohol dehydrogenase, abbreviated ADH, or acetaldehyde dehydrogenase, ALDH2. Certain allele combinations of ADH2 and ALDH2 can be associated with alcoholism. Certain combinations are associated with different regions. For example, ALDH2 alleles 1 and 2 and ALDH2 alleles 2 were lower than ALDH2 alleles 1 and 1 and ADH2 alleles 1 and 1, which were higher among alcoholic Korean men. As well, early onset of alcoholism in Mexican Americans were found to have a high frequency of the allele CYP to E1C2, a genetic susceptibility to fetal alcohol syndrome. In experimental animal studies where we studied the effect of alcohol, MAPK, TGF-beta, and the hedgehog signaling pathways were linked with deleterious effects. Fetal growth was affected among alcoholic women with the maternal genotype CYP17A1A1. Fetal alcohol syndrome can also be linked to paternal drinking. Paternal drinking can lead to the reduction of DNA methyltransferase activity within the sperm, resulting in DNA hypomethylation of sperm genes and subsequent activation of normally silenced genes. Hypomethylation can be transmitted through fertilization and affect the expression of genes in prenatal development. Altered gene expression can lead to fetal alcohol syndrome-like features in the offspring. Behavioral and developmental effects and alcohol exposure. The study assumes consistent alcohol exposure during each trimester of pregnancy This exposure includes being exposed to ethanol several times per week. There is a positive correlation between ADH and alcohol exposure found in 26 prenatally exposed children. There is a linear correlation associated with the severity of the disorder and the consumption of alcohol. Alcohol can affect intellectual abilities. Studies investigating the IQ value in the alcohol exposure shows a decrease in 7 IQ points given prenatal alcohol exposure. 9-year-old children that were also diagnosed with fetal alcohol syndrome were weaker in the reading, spelling, addition, subtraction, phonological awareness, and early literacy test. In the second semester of pregnancy, post-organogenetic period, the cerebral cortex is susceptible to the effects of alcohol. Children exposed to alcohol during each trimester showed cerebellar developmental disorders and a disproportionate reduction in the anterior cerebellar vermis. There is evidence of smaller frontal lobes and a decrease in choline concentrations when exposed to ethanol for prolonged periods of time during pregnancy. Alcohol exposure can lead to a decrease in size of the caudate nucleus. They also found a reduction in cerebellar growth and a reduction in cranial to body growth of the fetus. Cerebellar growth was normal if the mother stopped drinking at the beginning of the pregnancy. Binge drinking during pregnancy increased the rate of somatoform disorders, paranoid tendencies, substance-dependent, passive-aggressive tendencies, antisocial tendencies, and other personality disorders. So if you like this video, please like and subscribe. Join us for next time to uncover the criteria for fetal alcohol syndrome and organ abnormalities due to prenatal alcohol exposure.